Hello sir, myself Soham Surya. Welcome to my presentation that is coliform count as indicative of safety and quality. Now we should start from the introduction, that is coliform bacteria are also known as indicator organisms. Coliform refers to a wide variety of bacteria that can be found throughout the environment. This means that these organisms can be found in soil, water surfaces, vegetations, as well as on the skin or intestinal tract of warm blooded organisms such as humans, pigs, cows. Although some are pathogenic and some are non pathogenic. These are indicates in the presence of potential disease causing bacteria not only in water but also in given food and drink, that is milk. Therefore coliform are important because they help raise awareness and determine the source of the bacteria. Next, it can be divided into three main groups, which include total coliforms, fecal coliforms, and E. coli. Now, total coliform bacteria. This group is largely composed of harmless, closely related bacteria apart from humans and animal waste. Total coliform bacteria can be found in such environment as water, vegetation, food, soil, where they live freely. While, while they are generally harmless, the presence or detection of this group of bacteria in drinking water or water sources that supply drinking water is important because they are indicative of possible contamination. If detected in a water sample, this suggests that disease causing coliform may also be present, and thus the need to treat the water source or determine the source of contamination, that is environmental contamination. Next is thermotolerant coliforms are the good example of total coliform bacteria. These bacteria are capable of fermenting lactose at 45 degree. Detection of total coliform does not necessarily mean that disease causing bacteria are present in water. Testing the presence of total coliforms bacteria basically involves growing them in lactose media at 35 degree. Next is the fecal coliform bacteria. It is the subgroup of total coliform bacteria which found intestine and fecal of warm blooded animals. E. coli, it is the example of typically resides in the intestinal tract of warm blooded animals and thus the animal's fecal matter. When they are outside the host body, these organisms cannot live because it totally depends on the host body. Compared to total coliform bacteria, which are largely harmless, the fecal are composed of both pathogenic and non-pathogenic bacteria. As such, their detection in a sample of drinking water is an indication that the water is contaminated by sewage. The presence of these bacteria is also very important because the source of the bacteria is well compared to the source of total coliform bacteria. Therefore, it becomes easier to locate and fix the source of the problem and treat the water more effectively in order to prevent possible disease associated. Fecal coliform bacteria can also be found in such animals as shellfish, so people can get sick either by drinking water contaminated or getting eating contaminated shellfish. Some of the illness can cause mild stomach upsets to serve salmonellosis caused by salmonella bacteria. Next is E. coli. This is the whole figure of E. coli coliforms. E. coli is the subgroup of fecal coliform bacteria and is largely composed of E. coli compared to others. E. coli are almost exclusively found in the intestine of warm-blooded animals, where they are able to live and reproduce. Although they are mostly harmless in the host intestine, there are strains of E. coli. These are strain example E. coli 0157H7 that can cause serious illness. Detection of this organism is water water is indicative of fecal contamination. E. coli cannot live long outside the host. And now types of coliform, that is Citrobacter, Enterobacter, Hafnia, Escherichia, Clavicilla, and all types of coliform have genus in which is Citrobacter, a genus in Enterobacteriaceae family, Enterobacter a genus in class Gamma Proteobacteria that include Enterobacter aerogens, Hafnia also belonging to the Enterobacteriaceae family, and Escherichia also belongs to Enterobacter and include bacteria like E. coli. Some coliform characteristic. Coliform bacteria are gram-negative bacteria, and it do not form spores. They are facultative anaerobes. It means they can survive with very little or no oxygen by using anaerobic respiration. It is rod shaped, also known as bacillus. They are shaped like a rod, elongated. They ferment lactose. And now indicator test. The following are the some of the testing method used to determine whether total coliform bacteria are present in a sample of water. The first one is membrane filtration technique. In membrane filtration technique, our requirement is water sample and membrane filter. The filter used for the technique has pores of 0.45 micrometer and measures about 47 millimeters. MI incubator. Its procedure are pour about 100 millimeter of the water sample through the filter, place the filter on the plate agar on MI agar, incubate for about 24 hours at 35 degree. For this technique, the filter membrane is used to filter and thus retain any coliform bacteria that may be present in the sample. After incubation, the bacteria will use the nutrient in the agar plate to grow, if present. A blue color is observed. This indicates that the presence of E. coli. A fluorescence appearance, however, indicates that beta-galactosidase was involved in breaking down 4-methylubyl lifrel, which is also present in the agar. For this technique, absorbent pads with lauryl tryptose broth can also be used. These are transferred to either this media or agar to grow the bacteria. Once bacteria are cultured, the colonies are then counted under the microscope. Next is the multiple tube fermentation technique. To determine the presence of rod-shaped facultative anaerobic gram-negative coliform group of bacteria that do not form spores, requirement is lauryl tryptose broth glass tube. Procedure for this technique, the procedure involving three main phases: phase 1, phase 2 and phase 3. In phase 1, presumptive test, phase 2 confirmed test, and phase 3 completed test. We should start from the presumptive test. Add laurel triptose broth into several fermentation tubes. Inoculate varying quantities of the sample into the tubes. Incubate the tubes for between 24 and 48 hours at 35 degrees centigrade, and it's check every 24 hours for gas formation. Gas formation in the tube is marked as positive test. Phase 2 confirm test using fermentation tube with brilliant green lactose bile broth. For this step, only the sample that marked positive in the first phase are used. To the fermentation tube with the bile broth, inoculate a sample of the medium broth from the tubes that tested positive in the first phase immediately after gas formation. Inoculate the tubes for about 48 hours at 35 degree. If gas is formed during this phase, it is an indication of positive confirmed test. And last is completed test, phase 3. For this phase, requirement includes sample with positive confirmed test, eosin methylene blue plate, incubator, laurel triptose broth, fermentation tube, as well as nutrient agar slant. Using a wire loop, its procedure are using a wire loop, a scoop, and stick the methylene blue plates with the sample. Incubates the plate for about 24 hours at 35 degree. Obtain a colony of the bacteria from the plate and transfer it to the fermentation tube and nutrient agar. Incubate the two for between 24 and 48 hours at 35 degrees centigrade to determine whether any gas is produced. For the agar sample, staining is required in order to view the sample under the microscope. As for the fermentation tube, check for presence of gas. The presence of gas in fermentation tube indicates the positive test, and it marks satisfactory completed test. And next technique is MPN, means most probable number technique. Essentially, MPN is similar in principle to multiple tube fermentation technique. However, rather than simply being used to determine the presence of the bacteria, MPN is used to estimate bacterial concentration in water in order to determine whether the water is safe for use in homes. As with multiple tube fermentation technique, the technique involves three main steps of presumptive test, confirmatory test, and completed test. The sample is diluted in different tubes of different samples concentration and inoculated in lactose broth. These techniques makes it possible to determine the amount of bacteria in different dilution of the sample after they are cultured. The presence of bacteria in the sample is indicated by the production of either gas or acid, change in the color of media, or presence of bubbles. And last one is microscopy test. It can be used to view E. coli, coliforms in wastewater, groundwater, urine. Requirement is membrane filter, a pair of force, a low power microscope, sample like water, urine, wastewater, collecting apparatus, a funnel, prepared agar plate. Procedure: place the funnel on the collecting apparatus. The collecting apparatus may be connected to a vacuum system. Place the filter below the funnel so that it is between the funnel and the collecting apparatus. Pour the sample slowly into the funnel and apply a vacuum in the collecting apparatus until the funnel is empty. If some sample is still in the funnel, pour some sterile buffered dilution water to rinse it. Apply the vacuum to empty the funnel. Using a pair of hose, Carefully remove the membrane filter and place it on the agar plate in a petri dish. Mekongi agar, red violet agar, etc. Cover the plate with a lid and invert the plate. Incubate the plate for about 2 hours at 35 degrees Celsius. Using a low power microscope, observe the coliform conforms colonies on the plate using 10 to 15x magnification. Count the number of individual colony on the filter. To determine the number of colonies in 100 ml of the sample, divide the number of colonies counted with the millimeters of the sample used in the procedure and multiply the result with 100. This will give the percentage of the colony in the water, urine or wastewater. And last observation, depending on the type of coliform present in the sample, microscopy will show weak or high fluorescence intensity from the fluorescent probes attached to the bacteria. This makes it possible to count the number of individual bacteria in the specimen. Thank you, sir. Thank you, everyone.